Alright, so this video is for Biology 234 for the hands-on lab titled Hand Washing and Normal Flora. The learning objectives for this lab are to culture microbes from washed and unwashed hands, to categorize microbes by colony morphology and gram staining, and to analyze the effectiveness of hand washing. So we'll want to gather all of our materials first. So of the student supplied items, you'll need one agar plate. So these are my agar plates that have been in the refrigerator. So I'll just take one out and let that come to room temperature. And then put the rest of these back in the fridge. You'll need a bottle of bleach. You'll need a bottle of distilled water. You'll need matches or a lighter. You'll need your smartphone or a digital camera. You'll need 2 disposable cups. You'll need hand soap, which I have at my sink. You'll need isopropyl alcohol. You'll need a permanent marker. You'll need a roll of paper towels. And you'll need a source of tap water, which I have at my sink. And then of the hands-on lab supplied materials, you'll need your apron. You'll need blank slides. You'll need your face mask with ear loops. You'll need 2 inoculation loops. You'll need a pair of gloves. You'll need your staining tray. You'll need the tea candle. You'll need your test tube clamps. And then you'll also need your gram stain kit. Alright, so we'll move all of these items to the side and get started on exercise 1. So now we are ready to get started with exercise 1, which is hand washing and bacterial growth. In this exercise, you will inoculate an agar plate with bacteria present on your hands before and after washing with soap. Now we're ready to get started on the procedure. Step 1 is to gather the prepared agar plate poured during the Kirby Bauer experiment, a permanent marker, soap, and paper towels. So it does not say, but first I'm going to sterilize my bench top with 10% bleach. Here's my agar plate, and I'll need my Sharpie and then soap and paper towels. Step 2, use the permanent marker to draw a line through the middle of the bottom of the agar plate. So by that they mean the bottom of the dish where the agar is. So we'll just draw a line right down the middle like that, and this is the image that they refer to. Label one side pre and the other side post. So it should look like that. And then they also wrote soap on it, so I'll just write that. Step 3, open the plate on the pre-side and press your index finger into the agar for approximately 3 seconds. See figure 6. And so this is figure 6. So this is the pre-side, so it'll be on this side. And then I'll just open it to touch for 3 seconds. All right. So now we're at step 5. Wash your hands thoroughly with soap and running water as described in the background section. For hand washing, I'll just read what it says. The proper method for hand washing. Step 1, wet your hands with clean running water. Step 2, apply soap to wet hands. Step 3, create lather by rubbing the soap between your hands and scrub between the fingers, under your nails, and over the entire surface of your hands. Continue scrubbing for at least 20 seconds. Step 5, rinse your hands under clean running water. And then step 6, dry your hands with a clean paper towel. So we did this in a different lab with UV stuff that will show where you cleaned your hands and where you didn't. So I might include the results of that so that you guys can see, because that was kind of fun. But one thing I remember from that lab was that stuff gets stuck like in the creases of your knuckles. And then mostly stuff was getting stuck like in my nail bed. Like I seemed to focus on washing under my nails, but I didn't really focus on like the sides of my nails. And so that's where I noticed that the hand washing didn't quite get. So you'll scrub and then you'll scrub in between your hands. And then I usually will like scrub under my nails. And then now that I know, I try to make sure to like get my knuckles and the sides of my fingers when I'm rubbing the lather of soap before rinsing. So I'll go wash my hands and then I'll be right back. All right, so I'm gonna fully dry my hands and then so we're at step 5. Wash your hands thoroughly with soap and running water as described in the background section. Step 6, completely dry your hands with paper towels. And then step 7, on the plate on the post side, press your index finger into the agar for 3 seconds. So this is the post side, which will be on this side now. And then I will use my index finger for 3 seconds. So 1, 2, 3. All right, so step 8, remove your finger and return the lid to the plate. Step 9, place the inoculated agar plate in your incubation location, agar side up for 48 hours. And then step 10, after placing your plate in the incubation location, formulate a hypothesis related to the number and types of bacterial colonies that will grow on each half of the agar plate. Note, an example of a hypothesis is the pre-half of the plate will exhibit greater diversity, but fewer total colonies than the post side. And then step 11 is record your hypothesis in panel 1. And then the next step is to check your plate after it's grown for 48 hours. So I will see you guys in 2 days and we'll see what grows on my soap plate.